The mechanical properties of skin depend on structural systems that extend beyond individual proteins. This presentation examines how extracellular matrix organization and cellular regulation shape skin firmness, elasticity, and resilience over time. This content is part of the Beauty from Within series developed under the Natural Scientifics Research Framework and authored by Brian Geniuson. The material is presented exclusively for educational and scientific discussion purposes. Collagen is frequently described as the primary determinant of youthful skin. However, collagen does not function in isolation. Skin structure is governed by the extracellular matrix, or ECM, a dynamic and responsive network composed of collagen fibers, elastin, proteoglycans, and associated signaling molecules. Youthful skin reflects not merely the presence of collagen, but the integrity, organization, and adaptive regulation of this matrix. Dermal fibroblasts serve as the central regulators of extracellular matrix construction and maintenance. These cells integrate mechanical forces, biochemical signals, and metabolic state to coordinate collagen synthesis, elastin assembly, and matrix remodeling. With advancing biological age, fibroblast signaling fidelity declines, resulting in reduced matrix synthesis and impaired structural organization. As fibroblast regulation becomes disrupted, matrix metalloproteinase activity increases relative to matrix synthesis. Collagen fibers become fragmented, elastin networks lose coherence, and proteoglycan composition shifts. The extracellular matrix transitions from a flexible, load-bearing scaffold to a mechanically compromised structure with diminished elasticity and resilience. Importantly, increasing collagen availability alone does not restore extracellular matrix integrity if fibroblast signaling pathways and mechanotransduction remain impaired. Without coordinated cellular regulation, newly synthesized collagen may be improperly organized, rapidly degraded, or functionally ineffective. At the tissue level, extracellular matrix degradation manifests as dermal thinning, loss of tensile strength, reduced recoil, and impaired hydration retention. These structural changes present visibly as sagging, deepening wrinkles, and loss of skin firmness. From a systems biology perspective, extracellular matrix aging reflects a failure of coordinated cellular regulation rather than a simple deficiency of structural proteins. Within this framework, natural bioactive compounds and lifestyle factors that influence fibroblast signaling, redox balance, and cellular energy status represent biologically plausible leverage points for supporting extracellular matrix integrity. This discussion does not assert therapeutic outcomes, but instead outlines mechanistic relationships supported by molecular and cellular research literature. Validation summary. Mechanistic certainty for extracellular matrix regulation is high, supported by extensive cell biology and tissue mechanics research. Preclinical evidence from in vitro and animal models is strong. Clinical relevance is supported by observational and mechanistic human data, while translational readiness remains moderate due to the complexity of matrix regulation and inter-individual variability. Supporting scientific literature is available in the accompanying materials. This content is provided for educational and scientific purposes only, and does not diagnose, treat, cure, or prevent any disease. Highlight: skin structure is not maintained by isolated components, but by coordinated biological regulation. If you find this material informative, you may consider liking, sharing, or following this channel to support continued educational content.